Okay, hello everybody, welcome. This is Globins part 2. So throughout this video you should be able to describe the impact of allosterics, that's a vocab word, on oxygen transport, describe the T and R states structurally and thermodynamically of hemoglobin and their impact on oxygen transport, and then compare MB is myoglobin and HB is hemoglobin. Okay, so from last time we covered how iron is manipulated to better bind oxygen reversibly by adding it to the porphyrin and then adding it to the protein. This increases the reversibility and the solubility. However, this is still too strong. So for example, myoglobin, which has the heme and it's a protein, is not capable of reversible binding, so it's actually not very good at letting go of oxygen. And so we need one last piece of the puzzle in order to get efficient reversible binding of oxygen transport, and it turns out that's our friend hemoglobin, who is excellent at reversibly binding and solubilizing, because it's a globular protein, oxygen. All right, so structurally, what is the difference between these two? So this again is part 2. This is all about allosteric. So structurally, what's the difference between these two proteins? So here is a picture of hemoglobin. You can see that it has, let's see, what color should I use here? It has 1, 2, 3, 4 subunits. That makes it a tetramer. And this alpha and beta does not refer to alpha helices and beta sheets. Hopefully you can see that there really aren't any beta sheets. But the alpha and beta refer to two different primary structures. They're similar, but they're not identical. So that makes hemoglobin a heterotetramer. And I don't have a picture of myoglobin on this slide, but myoglobin is essentially only one of these. So it's a single subunit. It's always a single subunit. It's a monomer. So MB is a monomer always. So its highest level of protein structure is tertiary, and hemoglobin's highest level of protein structure is quaternary. So again, myoglobin highest level is tertiary. It's not great at reversible binding of oxygen, and not reversible enough to be transported through the bloodstream. But hemoglobin is really great. And again, the last piece of this puzzle is allosterics of the quaternary structure of hemoglobin. So let's get to that. So how does the tetrameric structure of hemoglobin influence reversible binding? So what I want you to see here is that hemoglobin is allosteric. So that is essentially the short answer to this question, is allosterics. Now what the heck does allosterics mean? Allo is a prefix meaning other, and steric is a prefix meaning shape. So a protein that is allosteric has multiple shapes. So what you'll see here, this is shape number 1, and this is shape number 2, and the difference between the two is that there's this 15-degree rotation between the subunits, and there's many ways to view this. This is just one of them. So because there's this movement of the protein, it has two shapes that makes hemoglobin allosteric. And we're going to talk about how the allosterics of this hemoglobin make it really excellent for reversible binding. And we'll talk about cooperativity and KD next time. So we can put some numbers to these things so you can see how reversible and how awesome hemoglobin is at reversible binding of oxygen. So the T state, this is the low affinity state, and the R state is the high affinity. I don't think I mentioned in the previous slide, but each subunit has a heme, one heme. So a hemoglobin has 4 heme groups. Myoglobin has 1 heme group. That means that in the low D state. So here we have hemoglobin in the T state. That should be a capital T. T state is unbound. There is an equilibrium where we can add 4 oxygens. There's an equilibrium to the R state, HB, which is oxygen bound. So 4 oxygens are bound, and we call this the R state. T and R refer to T meaning tense and R meaning relaxed. T and R are not specific to hemoglobin. It is these are general terms that you can use for a protein that has 2 conformations or 2 shapes, one being a low affinity binding and one being a high affinity binding. Just food for thought. So with this equilibrium arrow means is that there's an equilibrium between these 2 states. There's an equilibrium between the unbound state and the bound. Does that make sense? Maybe a little bit, maybe not. Okay, so if we increase oxygen concentration, we should shift towards the R state. That is Le Chatelier's principle. If oxygen concentration is decreased, then we should shift towards the T state. That is the unbound state. Again, that is Le Chatelier's. Okay, so again, we have a tetrameric structure that has allosterics. It has 2 shapes. It has a low affinity shape, a high affinity shape, and those are represented by the T state and the R state. And we're going to talk about what causes this change in shape, and then next we'll talk about what stabilizes each of them at the different oxygen concentration levels. So why does the quaternary structure even change? What is the mechanism of the allosteric effect? And it turns out it all starts with the heme group that is again bound to this. This is the proximal histidine. So it turns out unbound, so this is deoxymyoglobin, an oxymyoglobin, unbound, that iron 2+ that sits here is too big to fit in the plane of the porphyrin. It's too fat. Does that make sense? So it pops out of the plane. So in the T state, the heme is bent. So I don't know if you can see, there's a little bit of a bend upwards because the iron doesn't fit inside the plane of the porphyrin ring. Now once oxygen binds, it pulls some electron density away from that iron, and the iron slides into the plane of the ring. So hopefully you can see between these 2 pictures that this heme is actually relatively flat. And what that means is that the histidine, the heme moved down, and the histidine attached to it moved down. So in the R state, the heme is flat because the oxygen pulls electron density away from the iron. The atom itself gets skinnier and it slides into the plane. So the oxygen binds to the iron goes into the plane of the porphyrin ring. Now the proximal histidine is attached to the iron, so that slides down. And the histidine is attached to an alpha helix. So the alpha helices slide down, and the alpha helix is attached to the rest of the protein. So the rest of the protein slides, and what ends up happening is you've got these globins. I'll just do my SAD. Maybe I should have done them in different colors. There's 1 subunit, there's 2, 3, and 4. So when you get 1 O2 binding here, you get a change in the structure, the shape of the first one, and it actually causes a change of shape in the region between all the subunits where it gets, too many colors. The space in between here gets smaller. All right, I'm going to just explain that all one more time in case you need to hear it 2 times. So what is it that causes the quaternary structure of hemoglobin to change or to have allosterics 2 different shapes? So in the unbound state, where the heme is not bound to an oxygen, the iron 2+ is too big to fit in the plane of the ring, and so it's sticking a little bit out. Once oxygen binds, it pulls electron density away. That iron comes into the plane of the ring. The iron is then on the other side attached to the proximal histidine, which will also shift down. And the histidine is attached to a helix because it's the rest of the protein. And then that whole helice and that rest of the protein will also shift. Now, the subunits, as I've kind of tried to draw here, the subunits are also connected to one another. So when one subunit shifts, the next one shifts, and then the next one shifts, and then the next one shifts. And overall, what ends up happening is you have one state that has one shape, and you have a second state that has a second shape. And one of the defining characteristics between the two, in the T state, the area in between all of the subunits, there's like this big hole in the middle, is really large. And in the R state, there's just a teeny tiny area in the middle there. So next we're going to talk about what stabilizes each of the states. Okay, so what I have over here is the T state and the R state. And I'm actually, should I zoom? Should I not zoom? I have no idea. Let's just roll with it. So here's the question here, is what stabilizes each state? All right, so here's the T state. And each subunit you can see is in these different colors. There's these different colored blobs, and then there's these zoom-ins. These zoom-ins right here. So the T state is stabilized by salt bridges between subunits. Okay, so I'm going to have you focus on this one right here. So you'll see here that you've got this red residue on one subunit, the pink one, and then these blue ones on this other one. And in this diagram, you can see there's a number of salt bridges happening. These are things that are positively charged with things that are negatively charged. And then again, here you can see things that are positively charged and negatively charged having interactions. So the T state is stabilized by salt bridging between the subunits. And this can happen, or this is facilitated by, how do I want to say this? By the orientation of subunits with respect to each other. Okay, now in the R state, after the oxygen is bound, the heme goes into the ring, that pulls on the histidine pulls on the helix moves the protein, that shifts its relationship to the other subunits. Salt bridges break, but we form 4 coordinate covalent bonds and 4 hydrogen bonds. Gotta go all the way back. 4 H bonds. So what you can see in this picture is that the center cavity, that's what I should have said, the center cavity, I should have said that in the beginning one, the center cavity is smaller because the orientation of the subunits to each other is different. And so all the salt bridges that you had over here are now broken. However, you made 4 coordinate covalent bonds, 1 for each oxygen to its iron, and 4 hydrogen bonds for the oxygen to the distal histidine. So there's another way to represent this. And I'm going to zoom in here so that hopefully I can make this big enough so that you can see it. Okay, so we're going to do a reaction coordinate diagram. So this is reaction progress. I'm just going to remember reaction coordinate diagrams. They have energy. And they might look like this, or they might look like this, just so you can have a quick visual recall of reaction coordinate diagrams. We're going to do the reaction coordinate diagram here, except for we're not going to include the humps or the transition states. Okay, so reaction progress here, free energy here increasing. Remember, the lower the energy, the more stable something is. Okay, so in the T state, and then the other key piece of this is not meant to screw you up, but on the left-hand side, we're going to have low concentration of oxygen, and on the right-hand side, we're going to have an increasing concentration of O2. So oxygen concentration is increasing as we go along here in reaction progress. So at low oxygen concentration, the T state is lower in energy than the R state because of the salt bridges. The salt bridges lower the delta H of this, right? So every salt bridge releases energy, so we get a lowering of the enthalpy. Entropy is a wash here, it doesn't matter because the structure has already been formed. So at low oxygen concentration, the salt bridges are stabilizing and help keep it in that T state. Now, as we move towards high oxygen concentration, it turns out that the R state is lower than the T state, and the R state at high oxygen concentrations is stabilized by the 4 coordinate covalent bonds. This is between the heme and the oxygen, and then also you get 4 H bonds between the oxygen and the distal histidine. Now we have a cost, so this is all again decreasing enthalpy with the TH, enthalpy. This is all decrease in the enthalpy, which again makes it more stable. You had a cost, you broke a whole bunch of salt bridges, but that coordinate covalent bond sort of wins if there was a competition here. The coordinate covalent bond is much stronger than the salt bridges, so that overcomes that. So again, you should be able, come on, let me zoom out. You should be able to describe how each state is stabilized in terms of thermodynamics at the different oxygen concentrations, and this is only compounded by Le Chatelier's principle, HB T state, the unbound state, where we can have 4 oxygen molecules, which is an equilibrium, with HB which is bound to 4 oxygen molecules, and this is called the R state. So if we increase oxygen concentration, we should shift towards the right, towards the R state, and you should be able to describe what stabilizes that, the enthalpic contributions that stabilize that. If you decrease oxygen concentrations, we're going to shift to the left, where oxygen is now unbound, and you should be able to describe what stabilizes that state. Just to recap, so here we have the last little piece of the puzzle where hemoglobin's quaternary structure with its allosterics makes it an excellent reversible oxygen binding transport taxi, and hemoglobin is unable to do this. It's unable to do this because its highest level of protein structure is monomeric, is a monomeric tertiary structure. So hopefully throughout this video you've been able to add that little last piece of the puzzle and as to why hemoglobin is so much better than myoglobin at being able to transport oxygen through the bloodstream. If you were to switch them, if you were to put myoglobin in the bloodstream, it would soak up all the oxygens near the lungs, and then once it traveled throughout the bloodstream, it would just hold on to those oxygens and essentially asphyxiate all of your cells by not delivering or letting go of that oxygen. Whereas hemoglobin, if you put that back in the red blood cells, it will soak up the oxygen in the lungs and it will release it in the tissues where you need it, picking up the carbon dioxide back at the tissues and dropping it back off at the lungs, and we'll get to that in a later video. And I think I've talked too much, so I will see you in class. Bye-bye.